Okay, so just to show a quick moving model of what this would look like for the same cell going through either mitosis or meiosis. So you can see that just for 6 chromosomes, there are these pairs of homologous chromosomes that have hundreds of genes, but I'm just showing one gene where they have the same gene, but in this case, I'm showing the different allele to distinguish between the 2 different chromosomes. And then you've got a set that came from this cell's mother, and then a set that maybe came from this cell's father. So for either process, the first thing is going to be the copying of the DNA. And so the exact same chromosome, all the same genes is going to get copied. And these chromosomes don't have to be organized in any way in the cell. They're just making the identical copy. And it looks like I lost that one. Oh, here it is. So now, just to kind of show the big difference, if this is mitosis, then it's super easy. So they're going to all line up on kind of the midline of the cell, and they're going to split exactly in 2, right? You're going to get the exact same thing in both sides. So mitosis, they just line up directly in the middle, and you can see how when they split apart, the cells are going to get the exact same thing. If we're talking about meiosis, the first step is going to be the one that's the most different. So even before they line up, so again, we could copy them, but here is where the homologous chromosomes, so same genes but different versions, are going to come together, and this doesn't have to be in the middle of the cell. So this will kind of be, again, in the nucleus. These are going to come together and will swap segments. So you get a little bit of mixing up of genes from the maternal and the paternal chromosomes. But then as they cross over, they're going to get pulled to the middle of the cell. And the way that they line up is going to be with their homologous chromosome. And so that first division is going to be all about, first division of meiosis, all about separating the homologous chromosomes. And this is kind of the key step, that when they line up, it's totally random how they line up. So they could be like this, or they could be, this one could be switched. And so you can start to see how the way that they split here is going to be eventually how you get these different combinations in the gametes or the sex cells. So, this is one way that these could split. These split apart. These eventually split into 2 cells. And then eventually, when they split again, this is how you get, so this is meiosis 2. You're getting the replicated chromosomes splitting into 2. And then this is how you're ending up getting those 3 chromosomes in the gametes. And so we could do that a bunch of different times, and you would get a different combination every single time of these 3. And then think about if that were actually happening with 23 pairs of chromosomes, there would be a lot of different combinations.